Welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PI Collaborative Community aims to harmonize approaches to speed delivery to patients through collaboration in the pre-competitive space. The PI-CC is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law, because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection, since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda, and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of a discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward.
Here. All right, everyone, welcome back to the HER2-Low project with PI-CC. Again, as I mentioned, we're just giving Joe a few minutes. He should be joining shortly. And he asked that in the meantime, I just sort of go over what we spoke about in the last meeting. And it's all available on the website. I'm just sharing it here. If you weren't at the first session, we really just covered a broad range of topics, and then that led to a layout of potential topics to go into more detail with as the project continues. We did send around a survey for to find out what topics folks would like to focus on. I'll go ahead and share the link to the survey. If you didn't have a chance to select your preferred topic, if you don't mind just going ahead and doing it now, and then we could sort of look at what topics we should dive into more conversation about. And then also we did have a few folks who sent over some of their relevant work that might be interesting for others to look at. And it's all on the website. If anyone has any additional information or projects or papers that they would like to share, go ahead and send them over, and I'd be happy to add them. All right, so I'll just give everyone a few minutes to check out that survey. Again, these are the themes we were focusing on, and then we can look at the results to see what topics are worth delving deeper into.
I think clinically, the thing that we are really struggling with is, can we trust the test result that we're given? And how well does that test result actually predict benefit, because we have data certainly from DB04 that it didn't matter if you're 1+ or 2+. But I think in the clinical practice without central testing, a lot of times, obviously, one pathologist may read it as 0, another one as 1+. And, as the clinician, we struggle with, do we need to ask someone to reread it? Do we need to rebiopsy? And so I think this is a major challenge that we face. And I think will be an important thing to focus on.
Not anymore, in fact.
Yeah, and I think that looking at the results, it looked like one of the top choices was the topic on FDA on label versus LDT. And so it will be really interesting to see the next steps and whether this becomes something that the FDA wishes to take on to give you guys a little bit more guidelines when you're making these decisions.
And obviously all this can change if DB06 reports positive, because then ultra low becomes a whole other disaster. So we'll see.
Ula, you're muted. I don't know if you meant.
Oh, thank you. No, I was actually mentioning to Keith that he was muted when he spoke, and that's sort of ironic that I was too. We see you turn off the mute, but we still can't hear you. All right, so why don't we go ahead and take a peek at...
How about now? Can you hear me now?
Ah, there we go. Now we can hear
Okay.
you.
I think Sarah had mentioned a whole other, what did she say, disaster? I say it's a whole other opportunity.
Sorry, much better.
Yeah. A whole new challenge that we embrace and love.
All right, let me go ahead and take a peek at those results so we can see. And if any of these are the topics that folks suggested, maybe we can chat about them a little bit more. All right, so again, it does look like the top hits were that FDA on label versus LDT, an image analysis, and then acceptable range of discordance. So and then also,
Yeah, I will say some of these could be lumped eventually and turned into more, precise problem statements than they are. It was a great start, right? But
yeah, I mean, I think what we didn't get to last meeting, and what I think would probably be the most useful for moving forward, is again the conversation that we had in the page we put together is a useful resource, but it would be ideal to land on maybe a more concrete project that would have some impact for moving forward, and again, like hopefully regulatory science focus. And so I guess that would be, as Keith said, is like some of these topics can be lumped together and landed on something a little bit more concrete.
I mean, if you go back to the questions, a lot of the questions, for example, like differences over time and things like that.
This one?
No, the slide right before we had the poll. Right. I mean, you can't really ask questions about change over time and retesting and things like that until you sort out, or heterogeneity or anything like that, until you sort out like reproducibility. I mean, because if you can't reproduce either the reading on the same slide or the staining on the same slide, I mean, like, sort of pre-analytic or analytic, that kind of thing, then all of the other questions become unanswerable, because you will always have to say you're not sure if it really changed, or It's just the vagaries of testing. You know what I mean? I feel like that has to be the priority, because everything else is irrelevant if you can't show that your test is reproducible.
I do like the idea that we're getting an oncologist perspective here, because the outcome, what the pathologist does with the result versus what the oncologist does with the result they should be aligned, but they're fundamentally different, and the actions could be different if there's a lack of trust in whatever result is obtained, right? Based on the first shot. And a lot of that has to do perhaps not just with what assays use, but the lab practices, right? The countermeasures used in the lab to mitigate against errors, like, automatic consultations, things like this.
Joe, it looks like great timing. We were just going over the results of the survey and some of the topics, and a couple of good points have been brought up, is looking at the reproducibility of the test sort of before we get into all of what comes next. And then Keith brought up the pathologist perspective versus the oncologist perspective, and while ultimately they will be working together, there's some innate differences, and so having both perspectives on the call is very useful.
Very cool. So did everyone vote?
It doesn't look like everyone, but we got a lot of votes.
Nice. Very late. Sorry, just trying to find my way in where you are. So you went over the different themes, and we have some supplemented topics. It sounds like, right?
Yeah, I'll go ahead and share what were the takeaways. There's some stars on the topics that received the most votes, you could say.
Mm-hmm. Perfect. Yeah, I haven't seen this. LDT image analysis, an acceptable range of discordance. Does anyone know? I know we have a lot of experts on the call. Does anyone know whether a reproducibility study has been done with a specific aim on HER2-Low? I mean, I know that there are some, day-to-day variability studies and, reproducibility over time with older paraffin material. I've seen some of those studies, but
I mean, we've seen data we've seen data from David Rim's group where they did kind of like a round robin, just showing stained slides to a bunch of pathologists. I don't remember how many were in the round robin. And, for HER2-3+, there was pretty good agreement. I mean, it was very good. Between HER2-0 and HER2-1+, I mean, it was essentially like a coin flip. So that's the point I was making, that even if you assume like all the pre-analytical variables are correct and the staining is perfect, looking at the same slides, pathologists are classifying 0 or 1+ differently.
I think it was like a 26% concordance between 0 and 1.
So it really, and then on top of that, of course, depending on how much overstaining or understaining or whatever else, you could imagine you could also have some variation in the HER2 because the slide will look different. So like between those two factors, it's really hard to know how much to feel confident in 2+ different, but 0 versus 1+, I think it's very hard to feel confident as an oncologist. And speaking to the oncology perspective, we don't want to deny. Right now, we only can get TDXD covered patients who are HER2 1+. And so we don't want to deny patients the opportunity to receive TDXD, particularly because the small study called DAISY reported out, you know, a 30% response rate or thereabouts in patients who are clinically read as HER2-0. And so, but we can't get it paid for HER2-0. So at the moment, the sort of, for rightly or wrongly, at the moment, from an oncologist's perspective, we'd rather err on the side of calling it 1+ than 0. Now, you know, when DB06 reports out, we can sort of see like how important the thresholds really are and a little bit more about that. But I think that's the, you know, that's the current dilemma, is we can only prescribe HER2 1+. There's data for HER2-0, although it's quite limited. And we know that the concordance between 0 and 1+, at least based on the data from this round robin kind of exercise, is very low.
The other thing I would also bring up, because I think Nancy's exactly right, but then that's with the one time point biopsy, right? And then there's this beautiful autopsy series that shows that if you look at different spots in the same organ, that it's very heterogeneous, even at the same time point, in the same location. And then, you know, if you look across the body, obviously you also see this heterogeneity. And so I think it just makes this whole thing completely unreliable, because it's literally where you stuck your needle. Even if you could trust, even if we got the pathologist to read it the same, if we moved the needle over, you know, you may get a very different reading. And yet we're making a whole clinical decision based on this, which I think is a bit disturbing.
Right. And there is some reality to it in the sense that, you know, we and others have shown that, like, you know, ERBB2 amplification levels or RNA transcripts, I mean, they do you know, there is a correspondence between HER2-0 and HER2-low. HER2-low has generally more copies of HER2 and, you know, less ERBB2 transcripts, but it's overlapping, you know, Venn diagrams. So there is, I mean, it speaks that there is something real. It's not just totally lab artifact. Like, there really is something, but it's a continuum. You know, it's not bimodal.
So I'll just respond to what Nancy said earlier. I was one of the readers on the RIM paper, and I talked about this at the previous meeting. So apologies if you've heard this before, but when we were looking at the HER2 stain slides, there was no question of looking for 0 for 1+. So
Right.
there was no clinical relevance to distinguishing this. And so therefore, I strongly believe that now that there's going to be a lot better inter-observer agreement than there was before. There was also no standardization of how we would review these slides, even just to look for 2+ versus 3+. These slides were given to us as scanned whole slide images, and there's no standardization of monitor computer screen resolution, size, and the way that you approach looking at a digital image. It's just fundamentally different. So this touches on the reproducibility issue, but I just didn't want people to walk away thinking this study shows that we're really bad at agreeing. No. It's more that we're really good at focusing when we know what the clinical question is. And now that there is a very real, known, concrete consequence to reading 1+ versus 0, I can almost guarantee you that the inter-observer agreement is going to increase.
That also assumes that the pathologist realizes that it has clinical relevance, right? And I do worry about when you start getting into the community and in other areas, that may not be the case when they're reading it, because they're still following ASCO-CAP guidance, right? They're just gonna, you know, it's not like we've told them to change the way they're reading it, right?
Yeah, there's plenty of data to show that it takes about 10 years for any clinical guidance to make it into actual practice, regardless of what specialty you're in.
Yeah, thanks,
Would be
Amy, for
interesting.
mentioning that. Go ahead, please.
I was just gonna say, it'd be really interesting to look at even just the Brigham and MGH or whatever and see like what our read change is of how many 0 cases there are pre-approval of TDXT and post-approval.
Get right on that after I look at the ADH versus DCIS question.
So can somebody, maybe Amy, can you clarify, does CAP ask you what your fractional IHC bin scores are as part of your lab accreditation?
Yeah,
That's often used as a metric for, whether your assay's drifting and whether it's on target. Go ahead, please.
yeah, it does. And I don't have deep knowledge of it other than I participate in the CAP proficiency testing and inter-observer agreement studies that, our lab is part of. So we just had one and recently converted to digital review of these TMAs that they sent us. And in the past, it was just, everybody gather around a scope. So I also wonder about that component. Everyone's switching to a digital review of TMAs for HER2 scoring and proficiency testing for lab performance. Not sure what that's going to show. I'm sure they'll, report the results of that at some point.
Well, there's digital versus manual review, but then of course the scanners all have different image, tweaks that they apply to the image. If you take the same slide, scan it on 6 different scanners, there's a few published papers out there that show this. They all look great, like they all look nice. It's just that they look different. So if you're looking at a subtle membrane association of a cell, and if it's thin, you're wondering whether it's gray or brown. It could be more challenging depending on the scanner system, quality of the scanner, and just your bandwidth and your monitor calibration is a key piece too. It's a good point.
So it sounds like, despite the sort of topics that we had discussed last time, what we're sort of leaning towards is some of the pre-analytical variables that go into this change. And do we think that would be a more useful topic or area to focus on for conversations and a potential project?
I didn't realize, I missed the last call, so I think Amy's point is really a really good one. And, I think that paper is going to be quoted forevermore, despite the limitations that I didn't, fully recognize. And so I think, even if it's true that the community, you can't generalize to a community pathologist, it's still, I think, important. I mean, if, gold standard academic, breast pathologists can't get to a good concordance level, there's like no hope for community-based general pathologist. But if they can, then at least that says it is possible. And then it's a matter of training and implementation, all the other usual things. But, I think that seems like a reasonable, project. Because one of the problems right now is, Ana Garrido Castro and Paulo and a number of people in our group have, compared, for example, primaries and mets or residual disease and initial disease, looking at the HER2 scores. But if you can't be sure that there's, intra-test reproducibility, when you see a discordance rate of 20% or whatever it is, like every single caveat of every single discussion has to say, we don't know if this is really a change or it's just the scoring is not, it's just.
I mean, you probably all know from the original New England Journal of Medicine paper, there was a central testing, there was training prior to the test to reinforce the reading pathologists that that they've been trained on this 0 versus 1 distinction because it is important. and I think that was probably all. This is before I arrived at Roche, but this is all a requirement of the IRBs and the appropriate, the pharma company and all the stakeholders agreeing to the design of the trial. So just to keep that as locked down as possible. And I think it need to get detailed, updated information. But I think now on the Ventana side, the protocol has been locked. So at least depending on the geography, the protocol's been locked so that the lab can't adjust these factors in ways that either force a higher or lower score. I mean, they still can do that depending on what system they run, and this relates to the FDA versus FDA used on label versus LDT question. That's in the list. That it's up to labs as to what they do, right?
So I guess in terms of next steps, because I'm not sure I'm going to be able to contribute too much because I'm just not an expert on the topic. Do we think that there's a more useful question that we can ask, that we could share in a survey to see what sort of the community at large is, where their interests lie, and what next steps would be for continuing to potentially provide a resource to work on a white paper, or again, like address some of these reproducibility questions, the pre-analytical considerations, to get, as Keith just said, is like if every lab is going to be a little bit different. If it is an LDT, how do we build that trust between the oncologist and the pathologist, that they know what to expect with the reporting from the pathologist?
Sorry, Ula, I had to step out. Could you possibly repeat? I missed the first part of what you said. Sorry.
Yeah, no, that's okay. Well, essentially, I'm just trying to get to the next steps. Is like, unfortunately, I think Joe's even still having internet issues, and once he got to the lab, and so he's not going to be able to lead the conversation. And so I'm just hoping to get to how we can make this group of people productive, right? Is like, is there a different survey we should send out to the community? And if so, is there a specific question that we can ask them so that we know what direction to move in to create something useful? I think the conversations that we've had on the recordings and the collection of resources that's available on the resource or on the website is already a decent resource. But if there is a next step, it was pointed out, I think something that's sort of unique is the different stakeholders, right? We have oncologists and pathologists. Is that might be a direction to go, is how can we use that collaboration to create something useful?
One idea what I would have for a follow-up survey is, rather than asking a question generally about discordance rates and what rate would be acceptable, it would actually be very interesting to ask oncologists and pathologists separately, so you know who's responding, basically. What sensitivity for HER2-1+ would be acceptable, and what specificity would be acceptable? Meaning that, like, are we aiming for like 100% sensitivity for HER2-low? Like, we don't want to miss any. You know, if you do that, it's going to come at the cost of specificity, right? And sort of like, what's the lower bound of specificity that people would feel is acceptable? You're going to end up treating some patients who are actually HER2-0 with T-DXd. I mean, that, rather than just like, oh, is 10% discordance okay, or is 20% discordance okay? I think that would actually provide much better guidance for development of LDTs, and also much better guidance for pathologists. And if you end up with answers that are very far apart, then that's like a that's something that could be a subject of discussion, you know, that would be more focused as far as the output of what that discussion might look like. You know, you'd end up with some consensus about what is an acceptable performance of a test. I don't know. That's just some random idea.
I chime in here. I'm just trying to say, if we're trying to form develop a white paper as a task force, what are we hoping to share with the community? I mean, oncologists, pathologists, and is there any leading organizations like the CAP, ASCO? Are they doing similar surveys going on at the same time? So we're going to defeat the purpose. I mean, I'm just saying we have to come up with the why. Why are we? What are we hoping to do with this task force, and what's our intention, basically? Some sort of a guiding principles to guide us so that we can think about how we wanted to restructure the survey, but also develop the white paper.
I mean, it'd be nice to have some sort of guidance, particularly for clinicians in practice. I mean, right now we tell people that they can use any HER2-low assay from any time point to make a decision with regards to use of TDXD. Obviously, that didn't require any type of central review. That was just any reading over any time point, primary or met. And yet we know it's a dynamic marker, but we would need more data because in truth, we don't really have clean data that shows that if you have a HER2-0 reading as your most recent reading, and you were previously HER2-low, that the drug works as well if you were consistently HER2-low or HER2-low in your most recent reading. So we don't have the level of data to guide people here about that issue, nor do we have the issue, the data about using a local result, right? All the trials were based off a central test. And so I find it a little hard to know kind of, we would need data to guide people, and we just don't have that level of data, unfortunately.
Well, and maybe we could, as a group, call for that data, but we can't certainly organize, a study because it's that would have to be by Daiichi or by AstraZeneca or by an investigator-initiated study of some sort.
And there are actually a few trials that are ongoing and planned to sort of flesh out some of these things. And but I think from a more practical standpoint, I guess we could look at, if we are doing surveys, kind of we could show what's being done in practice, and we could ask people, are they using any read? Are they rebiopsying people? Are they asking for rereads? And then I don't know if it's possible to show, if we're able to pull from databases, what the change in reads has been over time, prevalence of HER2-0 versus 1+, 2+, like look at years pre-TDXD and post. I think that would be nice to show, because that would suggest an upreading is happening, which I think we all anticipate would happen, but again, no one's proven that is happening.
Yeah, I saw some data. Not sure if it's out in the hard literature yet, but at a recent meeting, the French groups showed that over time, they tracked this nationally, their distribution of HER2, and as soon as it was approved, the ADC was approved, like the number of classes significantly increased.
Yeah.
Not surprisingly.
All right, well, I think, and again, Joe keeps sending, please apologize to the group on my behalf. And he said he'll watch the recording and try and help us come up with some next steps, because I think, as was brought up, and it's a formal white paper where we collect a lot of data is just not going to be possible. As Keith said, that's an AstraZeneca size problem, not a PyCC type problem, but maybe at least some kind of commentary talking about why this would be valuable, or what we would like to see the sort as pathologists and oncologists coming together, what they both are really looking for. But I don't know, I agree that there's some good questions, and it's nice that we do have the community to do things such as surveys to get a little bit of an additional input for moving forward. But I think unless anyone has any other conversation points they'd like to continue, we can probably, oh, go ahead, Keith.
I was going to say, I have another suggestion. Maybe if we could make a task of this meeting, I don't think it'll take long now that we've had this discussion. If you could pop up the survey again.
Yeah, of course.
If we could think about, let's just say 3 major topics to discuss as a committee around next steps, but each of them needs a better problem definition. So, and I think some of the topics could be lumped in. So I can't see the other.
There we go.
Okay, I need to see
This one or the poll?
the poll, yeah.
There we go.
And okay, here we go. Yeah, so it sounds like assay reproducibility is an important consideration that can have several things rolled up into it, including assay choice, laboratory measures, laboratory countermeasures, LDT versus FDA, and then I guess ASCO cap would be sort of rolled under that as well. Are you taking notes on this, Zula?
I am, and there's
Okay.
a recording as well, so we'll go back.
Sure.
and
So
jot
I'm
down
thinking
everything.
for reproducibility, if that's a topic, that may be the
Okay.
first one.
Okay.
I think, and then several things could be rolled up under that. I do think, did somebody mention, I think image analysis is a good topic, maybe around, again, obviously can't talk about any platforms or specific uses, but like what are the requirements for image analysis, maybe around validation, how that's validated in an individual lab. How would it be useful? What level of feedback from the program would the pathologist need to be confident, using a number from a program versus their own training? And then the third one, acceptable range of, what was that? That's right around reproducibility, I think. So that would be lumped up in the one above. So if those are 2 topics, does anybody have any suggestions on like a third topic? And then I'm guessing with these, we would either you guys would do it or we could all think about it. Like, how do we make a problem statement that we could actually address as a group, either with a survey or the aggregation of more resources or whatever? Anybody?
I just had a sort of different comment, but I don't want to stop anyone else from answering Keith's question. Maybe if folks are just chewing on that, do you mind if I just pivot to something else?
Please.
So this something I wanted to ask Sarah actually, as to whether you have a sense of what the other clinical trials that are happening, like where they are in terms of their data, when do you think they would have results? So I think in terms of putting something out there, it's important to consider our timing. There are people who believe that this HER2-low issue is just going to go away because it won't matter. We're just going to give everybody an ADC, even if they're a 0, or if they're a 1+ at any point, whatever the criterion ultimately turns out to be. There are folks who just believe, based on their experience, whatever, that this is irrelevant. It's going to disappear. So if it's going to take 5 years to disappear, it's going to be different. What we put out from this group would be different than if it's going to take 7 years to disappear, or if it's going to disappear in 9 months. And I don't have a good grasp on what the clinical trial landscape is to have a sense. And I was wondering if, Sarah, you could comment on what you know or wonder about yourself.
So I think the next, so right now, obviously it's TDXD is approved based on HER2-low, 1+, 2+, and not FISH amplified. DB06 included a subset of patients who were HER2 ultra-low, so between 0 and 1. And that we are anticipating will report in 2024. That means that we could get an approval, an expanded approval for TDXD, not only in an earlier line, but also to include the ultra-low category late 2024 potentially. But in terms of HER2-0 and making that completely go away, they're only planned more investigator-initiated trials. So, for example, we're doing one with David Rim's group, from Yale, where they are doing a prospective study of TDXD and HER2-0, but it's a very small trial, just like DAISY was a small study. So there isn't a definitive study that I'm aware of that is planned that will completely allow a true HER2-0 patient. So I do think we're a ways away from that. Well, I agree, maybe it's true, maybe you don't even need to document HER2 expression. We don't have a way that we're going to get that indication in the near term. There are guidelines, just so people are aware, that are being developed for HER2-low based on DB04 data pre-DB06. So ESMO has one. I know ASCO has one that's going to be coming out shortly from both of those groups. So, I think they realize that what they say now will be different in a year if ultra-low reads out positive. But they don't think we're going to get to HER2-0 in the next, at least 2 to 3 years.
Okay, I think that's helpful to know the speed of what's happening in terms of generating the data that we feel like we lack to really provide guidance. And if that's the case, then maybe there's value in the short term for this task force to focus on the question of an ultra-low read versus a 0, because that's where the clinical criterion is going to be detecting any degree of HER2 IHC expression and how reproducible that is. Whether people feel that if we did a survey, if that would be something people felt they need to be trained on. It's a little bit different. It's much more qualitative and categorical than HER2 1+ and above, where you're dealing with a semi-quantitative component, something like that.
Do you think it's premature in a sense that we don't know if the drug will have an indication in the ultra-low? I think it's nice to start doing research on this area, but to provide commentary when we're not, I don't know, we're even sure if it actually works in that group of patients. It's a little early, but it's important to know, to prepare for, certainly.
Yeah, I'm not saying we go out and make some guidelines, but just to raise the issue that it's coming. And that even if this result doesn't show promise for particular drug X, that there's already precedent been set for an ADC being effective, and we're increasingly going to be asked to identify patients in order to submit them to these sorts of clinical trials to begin with. So even selecting that cohort you're going to examine, how reliable is that selection?
Well, it was all centrally selected, so we weren't asked to provide an example prediction of that.
Right, but then if you're going to roll it out, to some eventually to non-centralized labs and pathologists, there's that downstream effect, or the subsequent problem, challenge, opportunity, whatever you want to call it. Keith.
Yeah, I've got a couple of comments to follow up with yours, Amy. One is that we all know, HER2 is basically a prototype, right? It's like a paradigm for personalized medicine and selection of patients based on a single IHC test. So I think as you probably know, that's established a lot of the paradigm. It's established the operating principles around how we deal with these assays, how we standardize them, and how over 2 decades we've come to a point where we think we're good at a certain level, but we could always be better globally. So I think, and HER2, it looks like a drug, but there could be another one coming along in 6 months that's doing something a little different and still requires the same amount of testing, maybe a different threshold. We've got more outcomes coming out in the future, HER2-Low, as it's defined. I was in a CAP seminar last week, and one of the options, again, this is not a Roche thing, but this was mentioned as to all the pathologists in attendance, is that there may be need for 2 separate assays. One that's set up to detect very low levels, and one that's set up to detect the high levels that you get with the traditional 8 drugs that operate with HER2 positive. So I think whatever we decide to do, the situation is going to be transient. We don't know the time frame of these new approvals, but I guess one thing I would probably conclude from after having seen this field now for 20 years is that this testing isn't going away anytime soon, and it needs to be done reproducibly. It needs to be done fit for purpose, and so that is a principle I think probably we could all agree on. And the second one is that I think because of the maturity of the biomarker, the diversity of uses, it really is a paradigm, and there's a whole slew of other biomarkers that are waiting to be stained for and quantified, and pathologists read them, and then eventually image analysis or AI or whatever. So I think there's an opportunity here to put down some lessons and some things that we agree with that are applicable to other biomarkers. So I don't think either of the time is wasted, but I think focusing only on HER2 and only on HER2-low in the next 6 months it may be a mistake, because they're probably going to be changes. Thanks.
Yeah, I agree with that. And that complements well what I was trying to get at. But I would also add that, we have these artificial bins of 0, 1, 2, and 3, whereas when you look at ER and PR, it is a continuous reporting. It's a continuous variable, and there may be value for us to start reporting HER2 in a more similar way to ER and PR. And now that we are recognizing this as a clinical and a biological spectrum, and for this to be something, if we report it out in this way, we could reduce the number of times we're asked to go back and reread an old sample, because then this type of quantitative, semi-qualitative report can withstand the test of time against new requests for reads and trying to find patients to fit in a clinical cohort, etc. Or if you wanted to now go back and apply a new drug to someone whose specimen was read 3 years ago, you don't have to ask for a reread. Those criteria will persist, and they won't change, with regard to the bins that are created. Yes, you're probably still going to have inter-observer variability, but at least I think you're starting from a different place, and a better, more reproducible place, in a way. Do you know what I mean?
I think that's a nice idea. I guess the question is, how would you think about reporting it in a way that could sustain the test of time? It's funny, we were having this chat with Beppe Viale, who said he thinks pathologists need to report what percent of cells on the slide are 1 plus what percent, or 2 plus what percent, or 3 plus what percent, or 0, or whatever. There needs to be some type of reporting so you capture all the information. But how would you guys actually, what do you think is the right way to do that?
Yeah, I think that type of reporting should be done. It is that type of heterogeneity is sort of rolled into those 1, 2, and 3 plus categories. And I will say, though, that it is going to be challenging to ask a human being to do that. So this is where the AI component really can be helpful. I mean, we already have tools available to us that help us score HER2 as it is. So I think it isn't totally unreasonable to think we could have that in a somewhat near future. But like, it's not asking for a jetpack, but
Yeah.
but there needs to be also standardization against a reference. And typically a human being is used as the reference. And so I think that comes back to the whole, how do we validate AI machine learning algorithms? But I do agree that fundamentally we should be reporting the proportion of 0, 1, 2, and 3, because having that data point of heterogeneity will not only help patients get the right clinical treatment, but it will also inform future studies at all levels, whether you're doing it just like clinical pathologic correlation or whatever, or you're looking to enroll someone into a trial.
And that
Maybe
relates a
that
bit.
could be part of our commentary, if people agree, assuming people agree.
No, the point you make, Amy, is really good. I think then that relates to how you define reproducibility. If you're asking them to bin, in factors of 10, and your cutoff, but let's just say you don't even have a cutoff. Let's just say you're looking at distribution-related concordances across pathologists. Like, you're never going to get a homogeneous group that can distinguish 60% from 50%. Right? It's just not possible.
I don't think that
positive staining cells,
yeah.
right?
Yeah, I don't think that we don't need to have that per se. I mean, knowing that there's going to be variability. I mean, there's variability from maybe, oncologists don't want to know or want to hear about the variability we experience just staining one section today versus staining the same tissue section, like a different tissue section tomorrow. Your results are going to be slightly different depending on where in the operating or the dynamic curve you are for a particular marker. And so there's just inherently a variability from lot to lot of your antibody, from day to day, of your machine working, or the particular human being just doing the operating. And as a result, though, you're going to get inter-run variability, whether it's, machine driven, tissue driven, or human driven. But I think as long as we agree, this is coming back to one of the points earlier, is if we agree on what could be a reasonable level of discordance and disagreement, then that could help at least tighten the results for all patients. Like we have a similar problem in tumor infiltrating lymphocytes. You don't have people agreeing like, is it 30% or is it 40%?
Right.
But what matters more is that if you have, generally, if you fall in this range, that's good enough. And that may be also something that could be used in a clinical trial instead of a very hard cutoff of 50%. Be like, well, if you fall in this range, then that might be sufficient, because it should be a reflection of a real world operation, and not like an unachievable, not reproducible in real life type of setup.
I think for the purposes of registration, right, when you look at how these things are rolled out and used in trials, they do become dichotomous yes/no assessments, and these are often they're not based on H scores, right? They're based on a percent positive, let's say, above a given cutoff, and that cutoff is a bet, right? It's a bet based on the risk to benefit ratio of the drugs and the drug in each of those categories. So likely responders, likely non-responders. The way the FDA wants to see this is in positive percent agreement, negative percent agreement tables, right? So that I think translating whatever we're talking about into that format is how the regulators see it right now. And my understanding is that if you're anything below about 85%, it's not going to work, right? You need to be better than that at a minimum for most tests.
I have what might be a silly question, and I apologize. I'm a student to all of this. But this subjectivity that everyone is discussing around like IHC results and reporting, is that exclusive to HER2-low, or does that go across the HER2 reporting spectrum?
I mean, I guess the short answer is the focus of the test for the first 20 years of its existence was HER2 positive versus negative, and negative was anything that was either a 2+ FISH negative, a 0, or a 1+. So there really wasn't much incentive, as Amy mentioned, for a pathologist to spend the time to really make the hard call of whether it's a 0, or a 1, or a 2. There are, I'll call them textbook cases where, you or I or Amy could look at them literally in about 30 seconds and make the call. Getting this data out where you're looking at the bins, the percentage in each bin, that would take most proficient pathologists, I would bet, depending on the sample, 5 minutes to do consistently. And some of them don't have the time to do it if it's an open and shut case. They just don't spend the time. I don't know, Amy, do you have any additional? And I guess the last point is that like all the biomarkers have this issue. Every IHC biomarker has similar issues. That one easy and hard thing about HER2 is that it's in the membrane. ER and PR are in the nucleus. PDL1 is another one that's tested. Different biology, but you're looking for scoring membrane as well. So there's some of the same issues. Amy?
Yeah, I mean, there are similar issues in other biomarkers, and it really just depends on if you're using it for selecting treatment. It really depends on how the clinical study was set up to begin with, and then that defines how you end up utilizing it and interpreting it in real life on real actual, prospectively patients. So if you're dealing with a membrane marker, then they will have defined, what is the minimum that you need to see? Does it have to be complete or incomplete, the intensity, things like that. So these things have been generally defined pretty early on before they become clinically relevant. But when you start to implement it, you run into problems the more definitions you have. Like PDL1 and lung cancer, for example, is extremely I would much rather read PDL1 than read HER2, because PDL1's criterion for clinical treatment is 1% of cells with any staining on the membrane. And I'm like, oh, that was easy. And then I have a tray of HER2 and it's just like, oh, this is going to take me forever. And so the way that it gets rolled out into actual practice is important, because you also, there's a very real pathologist fatigue component to looking at a tray of PDL1 versus looking at a tray of HER2.
Well, but if you were in a reference lab where half of your PDL1s were one indication, one assay, and the other half were another indication, other assay, and you have to shift, your scoring, your mental scoring system, that's a burden for sure.
Yeah. So, this, I'm happy you brought that up, because I meant to say this earlier, that if we decide to transition to reporting HER2 proportions of 0, 1, +2, +, whatever, it actually would probably serve all the other patients who are not breast cancer patients, like GI cancers that are HER2 positive, and you have to run HER2 on those. Those are different criteria than breast criteria. I don't even, I don't do that regularly, but I know enough that it is a separate system, and therefore it would be helpful to all of us, all as in, all people who are patients and all people who are doctors dealing with these sorts of decisions, that we report things out in a continuous as a continuous variable, and then you can just re-bin people as enrollment or drug criteria change.
Great. So we are at the top of the hour, so I do want to be cognizant of everyone's time, and thank you so much for the great conversation. And again, apologize, Joe was not here to lead the meeting, but we will review the notes and the recording and sort of land on some next steps. I think again, this task force has a lot of potential, and landing on something concrete that we can contribute on this topic for moving forward would be very useful. So again, thank you all, and see you next time.
Thank you. Bye-bye.